So I'm going to talk about genome structural variation, and in particular, the particular class of structural variation known as copy number variation, that the form of structural variation that we know most about today. On some level, aspects of an appreciation that aspects of genomes can vary in copy number is actually not a recent revelation. So this is, for example, a karyotype from a patient. Can anyone tell what this patient's condition is? Yeah. So this patient has 3 copies of chromosome 21 instead of the expected 2, and that is that has a rather severe set of phenotypes associated with it, although varying in severity from patient to patient. But this had actually been recognized for many decades as the cause of Down syndrome. Anyone know what this patient's predicament is? Anyone want to guess? So cancer. This is a karyotype from a tumor, and you can see that it's evidence of really substantial genome instability in which it's acquired extra copies of certain chromosomes. There have been translocations and recombination events between chromosomes, giving you sort of longer and shorter versions of the standard chromosome. And similarly, it was recognized through analyses like this, which is really, this is not a molecular analysis per se. This is analysis under a microscope. It was recognized for decades from analyses like this that copy number variation was a feature of tumorigenesis. And so this is another more specific one, but also associated with cancer. This is called the Philadelphia chromosome, involving a translocation between chromosomes 9 and 22 in the BCR and ABL genes. But we tended, up until about 8 years ago, we tended to think of things like this as being features only of sort of cancer genomes and sort of congenital anomalies, like situations involving phenotypes that you could recognize practically from birth. And the sort of path toward realizing that copy number variation was much more common than that and extensive sort of went through an approach called comparative genomic hybridization. Now, today we think of comparative genomic hybridization, or CGH, in terms of array platforms. But actually, before array CGH, there was CGH, and it used to be done both clinically and in science. To do the following kind of experiment: you would take a karyotype from a normal person, and then you would take DNA from 2 other people. You might take DNA from a tumor and label it in green, and DNA from a control sample and label it in red, and then competitively hybridize it to this karyotype. And then most of the chromosomes would be yellow. They'd be bound by similar amounts of DNA from both samples. But then you could see these regions that were green or red that represented chromosomal sections that had been amplified or lost in the tumor. But the real breakthrough toward realizing that there was much more, that copy number variation kind of went beyond cancer and congenital anomalies, was really a set of experiments that were done by Johnny Afraidy when he was a postdoc in Charles Lee's lab here at Brigham and Women's Hospital, and that were done at about the same time by Jonathan Sabat when he was a postdoc in Mike Wigler's lab at Cold Spring Harbor. And these were actually experiments using CGH, but now instead of doing it on a karyotype platform, by making arrays of large genomic clones, typically BACs and phosmids, so clones that were themselves 40 to 200 kilobases in size. And this, but essentially doing the same experiment where you take DNA from different samples and competitively hybridize it. And because the size of these clones was much smaller than the sizes of things you could visualize microscopically, it made it possible for the first time to detect copy number differences between genomes that were what we call submicroscopic. And Their size wasn't known at the time, but presumably they were on the scale of like tens to hundreds of kilobases. And the really surprising observation that John made was that this phenomenon went far beyond cancer, that he could take genomes from different apparently normal people and competitively hybridize them to these arrays. And almost whenever he did that, he would see at least one or a small handful of regions that appeared to have be substantially different in copy number between different people. And so this made it clear for the first time that this might be a more common and extensive feature of human genomes than people had thought. Now, it is important to remember something different, though, about the sort of transition in approaches and ideas in the field at this time. Okay, so before, copy number variation was ascertained cytogenetically. So what people were seeing was it was by definition enormous. You had to be able to see it under the microscope. So sort of enormous on genome scales. It was usually functional, and in part because it was almost always ascertained in patients. So you didn't just go do karyotypes on like everyone. You would do them on people who had something really wrong. The sort of a transition in as these array-based technologies became available, which is that it became possible to start looking at these in just sort of large cohorts, but this wasn't always appreciated at the time, but that actually meant that the kinds of things that you might be looking at might actually be very different. So for example, now as you start ascertaining in large numbers of normal individuals, it becomes possible that many of these are benign. And that also becomes more possible because we're now looking at things that are much smaller. And so unlike these, which are sort of conspicuous and causal of phenotypes a large fraction of the time, with these it was really very much a question of like, how important are these? And so what was needed as of about 6 years ago was the ability to begin to relate this form of variation to common and complex disease. And at that time, genome-wide association studies were sort of first getting off the ground here and other places. And they were really the first times where people had assembled really large cohorts of individuals with different phenotypes and were sort of preparing to analyze them on SNP arrays. And so there was sort of an effort here at that time that was actually really sort of three trainees here. So this is a project on which I worked together with I was a postdoc then and working together with Finny Karuvula, another postdoc, and Josh Korn, graduate student. And what we did was we basically redesigned SNP arrays and analysis algorithms so that they so that you could sort of adequately analyze copy number variation in the course of doing these genome-wide association studies. And so in part, one of the reasons you needed to redesign SNP arrays was that they hadn't really been designed with analysis of copy number variation in mind. So we sort of populated them with more equally spaced assays that would give you access to all the different genomic regions. In fact, there was a phenomenon at the time where regions that were commonly affected by copy number variation were actually systematically filtered out of SNP assays by quality control. So we had to kind of go back and sort of fix those kinds of things and redesign them. And then within a couple of years, it just sort of became routine, like that. Whenever you did a GWAS, it would sort of also be analysis of copy number variation. So what did we and others learn from genome-wide arrays about copy number variation in disease over the following years? What this was enabling was really recognition and kind of widespread testing of copy number variation in common complex disease. And really the first findings really came in autism. For example, a paper by Jonathan showing that several percent of autism cases actually have a new deletion or duplication that they didn't inherit from either of their two parents. Really the first discovery using these SNP arrays that we had redesigned for analysis in GWAS was the discovery here by Lauren Weiss and colleagues that there's a recurring microdeletion, microduplication syndrome at 16p 11.2, and that several percent of autism patients had, or I think 1 or 2% of autism patients had deletions or duplications of about a half megabase in size at this locus. And you know, these observations were actually really quickly replicated, and then within a few years, actually, as sort of sample size increased in the field, it became recognized that there were actually several loci like this, at which there were recurring deletions and duplications in patients. And today, in addition to these loci, I'll actually get back to this in a second, but in addition to these loci at which there are recurring events, I'll get back to this in a second. So the same thing's also true in schizophrenia. So a year or so later, a group here, actually including most of the people I mentioned before, Josh Korn and Sean Purcell, and doing analysis for the International Schizophrenia, found that the same thing was true in schizophrenia as in autism, which was that there were several loci at which there were recurring deletions in on the order of 0.5% to 1% of cases. Yeah?
Can you compare the back hybridization arrays to the genome-wide SNP arrays in terms of detecting these copy number variations? What is the size? What is the resolution that you have in each one of them?
Can you repeat the question?
Yeah. So the question is, could you compare SNP arrays to back arrays and CGH arrays for the size of events that you could detect? So the back arrays, it turns out, we're often detecting events that were maybe even as small as 20 kilobases, which are also detectable on the SNP arrays. I think comparative hybrid CGH has really switched from using back arrays as a platform to using oligos as a platform, and so then it's just really a question of like at what density oligo array you use. These events are actually large enough that they were detectable on almost any platform. The reason that almost all of them were detected on SNP arrays was that was the technology that was really being used to look at kind of large cohorts for common complex disease. And so if you could sort of package it together with another experiment, it just made it sort of much more scalable to large cohorts.
What is the lower resolution limit that you have with the SNP arrays to detect CNVs?
So you can detect events as small as a couple of kilobases, but the question is, what is the size of events at which you can detect with the level of sensitivity and specificity that enables a rigorous genome scale analysis that will not be sort of inflated by technical things? And really on all these array-based technologies, unfortunately that size today is still in the high tens of kilobases. So it's not that smaller events are not detectable, but the question is, what's the size of event that's detectable in a rigorous analysis that won't be confounded by technical artifacts and things like that? So events that's detected with very high sensitivity and specificity. And unfortunately, that's still the really big things. Sequencing, whole genome sequencing will change that, and I'll get to that at the end. So one thing that was seen in both schizophrenia and autism was that in addition to these specific loci at which there were recurring events in many people, that there was just a general tendency that cases were more likely than controls to have a really large event somewhere in their genome. And by really large, I mean typical threshold might be 500 kilobases or more. So if you think of, say, deletions or duplications, 0.5 megabase or larger, that are rare but genic, those kinds of events are seen more frequently in cases than in controls. So maybe in about 2 to 3% of controls, and in about 7 to 8% of cases with autism, the case control difference is a little bit more modest in schizophrenia. And so this kind of result is called a sort of is often referred to as a burden, because it's sort of not, it's not about any one locus in the genome and connecting that to a disease. It's a more sort of pervasive, diffuse result, which is that these events are sort of more common anywhere. Our interpretation of this is that a subset of the events that are observed in cases actually matter, and an unknown subset actually matter, and an unknown subset are just kind of part of the general background of these events that are present in populations. So a few things that these recurring CNVs that recur at specific loci have taught us, and this is a table from Sean Purcell that that summarizes results at 7 loci at which recurring CNVs associate with risk of schizophrenia. So a few things I want you to point out. First of all, these events are all large. So these are all half a megabase or larger, except for these ones. And most of them affect many genes. That's actually made it very hard made the biological follow-up of these events really tricky. We still don't know what is the gene within these regions that is responsible for the phenotype. And there's not yet any result in which the effects of any of these deletions is phenocopied by a fine-scale mutation in one of the genes within the region in any clean, obvious way. The other thing I'd like you to notice is that each of these events has also come to be associated with other phenotypes. Okay, so they maybe were discovered initially in the context of one phenotype that was being scanned with particularly large sample size, but then as people went back and looked in other disorders, they would also see an enrichment in cases relative to controls. And initially, this actually surprised us a lot. So I remember when we were first working on the ISC paper and the CNVs in the recurring CNVs in schizophrenia, and one of them was were these deletions at the 1q21 locus. And at the time, in that cohort, we had only seen them in cases and not in controls. And it was easy to sort of exuberantly think that these might be almost sort of fully penetrant causes, especially because they are large, they're large events that are affecting so many genes. And it just turned out that it was because we hadn't scanned enough controls. And actually, a few months later, Josh Korn and I were working with Cricket Seidman and John Seidman, who were doing a similar study, but of a totally different phenotype, which were congenital heart malformations that are ascertained in newborns. And we found that those cases also tended to have, about 5% of those cases also tended to have CNVs at the exact same locus. And so we had not seen them in any controls in the schizophrenia study. And yet, in this study, we were seeing them in patients with a different phenotype, but who had a lot of these were actually now adults and had no psychiatric phenotype. And so it actually suggested that although these were, it taught us that although these events are present at substantially lower frequency in controls than in cases, they're not absent from the general population. They are statistical risk factors, but they are not fully penetrant. And by today, as the sample size for all of these things has increased so much, and these are being ascertained in all kinds of large studies, it's much more straightforward to estimate an odds ratio. You can see these odds ratios still have wide confidence intervals around them because they're still relatively rare events for which the numbers aren't high enough to really precisely measure an odds ratio. But the thing that I want to point out is that although these odds ratios are high, these are large risk factors, schizophrenia is an illness for which the background frequency in the general population is about 1%. And so even an event that confers that brings let's say this duplication that brings an odds ratio of 25, that may only increase your risk from 1% to 25%. So that still would mean that the majority of carriers in the general population might not have any psychiatric phenotype, or at least any psychiatric diagnosis. And That's important to realize, really not just to think about, not just about copy number variation, but also just about sort of rare variants in general that'll now be ascertained in the course of like exome sequencing and genome sequencing studies, which is that there will be a tendency to first see these in cases and to assume that they're like large, overwhelming risk factors. And one of the things that CNVs have taught us is that frequently these are much less overwhelming risk factors than we think, and that we really don't know until we've ascertained large numbers of controls. And there's a sense in which CNVs have just been a sort of a kind of leading edge, I think, just sort of a leading edge of just sort of about lessons about rare variation in general, that a lot of the things that these deletions, that I think when we look back at copy number variation, one of the things that we'll remember about it is just that it's the form of rare variation that we could see and start to think about first. It was the form we could start to see on array-based platforms several years before we had the technology to systematically ascertain rare point mutations via sequencing. And so these are kind of in broad strokes, I think, what some of those lessons are. Things that are strong risk factors, but not fully penetrant, not Mendelian, and that are pleiotropic. So they seem to have actually different phenotypic outcomes in different patients, potentially due to differences in genetic background in different patients. But it could be genetic background or environmental influences, things like that. And I think those things are likely to turn out to be true about rare variation in general.
So I'm just wondering how you compare the cases and the controls. So you see this recurrent deletion, and then you have a consensus of that deletion, basically the intersection of all of them.
Yeah.
Then you go to all the controls, and you don't even see, even if you in the middle like a small deletion inside, you never see something like that. It's completely intact in all the chromosomes. Like not even a small hit inside the big one that you have over there.
Well, it's not that you never see inside other, but
okay.
Yeah.
But how do you count if you see like a small deletion inside like the big consensus region in the control? Do you count it as a hit, or you need it to be the same size? Or how do you do this?
So this scoring is in terms of sort of the canonical event. You could imagine going back and saying like, okay, now are there other patients who have sort of smaller deletions within the region, and try to implicate specific genes within the region that way? And a lot of people have tried to do that, and it has not been successful yet. Not because my assumption is that in many of these loci, there will turn out to be an individual gene for which finer scale mutations, either smaller deletions or point mutations, more or less phenocopy the effect of the big event, but they haven't been found yet. Okay. And I think the reason is that there's something special about all of these recurring events, which is that they're at sites where they're flanked by homologous sequences, and it tends to be a specific event that recurs at higher frequency, whereas these smaller deletions at specific genes within the region are much more they're much more sporadic. They don't recur with the same frequency, so there has not been the numbers that would really allow you to say anything conclusive about them. Even the conclusions about these loci, the number of events that they're based on is in the tens. It's not the hundreds or the thousands. It's enough to establish these patterns. But now then you start to think about other events that have a much lower mutation rate. There just haven't been the numbers to learn anything yet. So, what have CNVs taught us about psychiatric illness? And I don't mean to see all of this through the prism of psychiatric illness, but psychiatric illness is a set of phenotypes in which it's really the set of phenotypes in which CNVs have been most strongly implicated, and through the prism of which we've sort of learned the most about them. So a few percent, on the order of about 5% of cases of autism and schizophrenia have a large de novo CNV that we believe contributes to their pathogenesis. This result of this sort of more diffuse genome-wide burden of CNVs in patients, one of the things that's taught us is about high polygenicity of these disorders, so that beyond these loci with the recurring events, the fact that there's this general pattern where CNVs at many different loci in the genome are large CNVs at many different loci are more common in cases and controls, tell us that these are highly polygenic illnesses. There are not results like this for diseases with a much more sort of simple, less polygenic architecture. And that tells us that there are probably hundreds of genes that can mutate to an influence on these phenotypes. And that then becomes a lesson that we then take to think about how do we do all the other complex disease genetics? How do we think about strategies for GWAS and sequencing? Because if we thought these were the kinds of disorders where there might be sort of 7 genes that can mutate to the phenotype, you'd employ totally different strategies than you would if they're the kinds of disorders where there might be hundreds of genes that mutate to phenotype. And CNVs have taught us that in particular, that autism and schizophrenia seem to be in this latter category. And again, not all diseases are like that. Your metabolic illness is not like that. You might have a pathway with 7 genes, and maybe any of those can mutate to that metabolic illness, but there aren't hundreds of genes that are pushing someone's risk around. So they've taught us about the complexity and polygenicity of these diseases. They've taught us about this space of alleles that they're not Mendelian causes, but nor are they alleles of weak effect. Taught us about pleiotropy, but individual genes and therefore biological impact have been slow to emerge, mainly because these regions implicate many, many genes, and large meta-analyses have been technically challenging. So I want to talk briefly in the next 10 minutes or so about some areas that are sort of frontiers in structural variation research today, and then let's take questions for the last 5 or 10 minutes. So one of those involves more complex and multi-allelic forms of structural variation. So the structural variation, the forms of structural variation that we know most about today are, and that have been analyzable in large cohorts via array-based platforms, are simple deletions and duplications, in which, for example, as you sort of march along a chromosomal segment and look at sort of hybridization intensity to a series of probes along that chromosomal segment, you might see a clear segment over which some people have greater hybridization intensity than others, suggesting a duplication, or less hybridization intensity than others, suggesting a deletion. This is sort of what the currency of data in this field has looked like, at least when it's clean. It's unfortunately seldom as clean or precise as that. So most insights today come from simple deletions and duplications. And in particular, today I've emphasized these results from rare deletions and duplications. But there's also actually a whole other universe of common deletion and duplication alleles that are just part of the reservoir of inherited genome polymorphism. They're generally smaller than the really large events that I was talking about just now. And they're mostly ancient mutations. And actually, in work that we did over the last several years, we showed that the vast majority of common copy number polymorphisms are segregating today on specific haplotypes, and that you can capture them by linkage disequilibrium to specific SNPs. But this sort of one slide summary about what we know about copy number variation in disease is really not about the whole iceberg of copy number variation. It's about these sort of simple deletions and duplications that you could ascertain on the technology that we've had over the last several years. But it was sort of clear all the time, we just never had the tools to analyze it, that there are also regions of high apparent structural complexity in populations, in which when you look at data like I showed you on a couple of slides ago, but now in sort of many people in a population, you'll see kind of all kinds of different patterns across that region. And it's assumed, although it had never really been shown, that what's causing data like this is that there's many different structural forms present at the same locus. Because it's always really hard to know what was going on in these regions because the data was really so ugly, you couldn't look at that and tell what was happening. And these regions were not genotyped. There was no clear way you could tag them by SNPs. You'd have to actually, first you'd have to type them to even know if you could tag them. And so they sort of always got left on the table in every study that was done, GWAS, exome sequencing. And so one of our goals has been to develop a set of molecular and statistical methods to analyze these structurally complex regions that harbor what we call extreme structural variation. And one example of that, and this is actually a paper that was published by Linda Betker and Bob Hansaker to a student scientist in my lab in Nature Genetics a few months ago. One example of a locus like this, which is sort of I think an initial proof of concept for unraveling complicated regions like this and figuring them out. What Linda and Bob and Mike showed was that this locus actually segregates in human populations in 9 different structural forms, each of which is characterized from all of the other structural forms by the gain or loss or rearrangement of more than 100 kilobases of genome sequence. So these are not subtle differences between these different structures. These are dramatic, large-scale differences. This involves the inversion of almost a megabase of sequence. This involves this sort of tandem duplication of a couple hundred kilobases of sequence. This involves the actual dispersal of another 150 kilobases of sequence from here over to here. And this is just sort of an initial proof of concept. I think this is something that over the coming years we can sort of make possible to do. We can take these from being sort of dark corners of the human genome to regions that we can think of as like, this is the set of structures that are present in the population. This is how they sort of formed from one another, the kind of mutational history of the locus. And then we can begin to write the playbook for bringing these into studies of complex disease. So for example, Linda and Bob showed that actually each of these structures can be imputed through flanking SNP haplotypes. And in fact, Linda's already gone on to now analyze these structures in a set of different diseases, such as Parkinson's disease, in which that had associated a genome variation at this locus, but for which the sort of causal alleles or haplotypes were unknown. I think one thing that may also turn out to be true of some of these loci is they just show very interesting evolutionary histories in human populations. So for example, at this locus, Linda and Bob showed that actually some of these structures are specific to Mediterranean populations and not seen elsewhere in the world, and that they've actually risen to high frequency in those populations within a very short period of time. So another sort of frontier in structural variation research today that I think is really important is going to be to go beyond these really large deletions and duplications that you can see on array-based platforms to ascertaining the full spectrum of structural variation that's present in each genome, and really using all structural variants, large and small, as discovery tools. And I think that's important because as much as we learn from these giant structural variants, there's far more small ones than there are large ones. And it's just always been, I think, a thorn in the side in the field that although it's clear that there are far more of these small ones than these large ones, it's been impossible to do really rigorous association analyses of any of them because they were always in this zone where they're sort of just barely detected, and the same event might be detected in one sample and not detected in another. And they've never sort of moved into this stable of things that people can do sort of high quality genome-wide association testing on in the way that we can of SNPs and now of rare sequence variation. And so a really important effort is to fix that. And so again, as much as we've learned from large CNVs, I think there's probably far more that we can learn from smaller structural variants. They'll tend to affect individual genes and exons, so we'll know what the functional genetic element at that locus is. And the problem's just that they've been hard to detect in a rigorous way. So I think what is going to allow us to turn the corner on this is whole genome sequencing. And in the last few slides, I just wanted to sort of give you an appetizer of what I think that future is going to look like. So structural variations leave several kinds of footprints in whole genome sequence data. One of those is the sort of abnormal separation of the read pairs that are sequenced from the left and the right end of a fragment in a sequencing library. So a sequencing library will tend to have a certain expected insert size distribution. And when you see pairs of reads that are abnormally separated, that can indicate the existence of a haplotype on which the intervening sequence is actually missing. Similarly, you can see there will be individual reads that fail to align, and then it turns out that that's actually because part of that read is on one side of a structural variant and the other part of the read is on the other. And finally, there's local variation in read depth. So if you look at profiles of read depth across the genome, it's not uniform, and there's all kinds of technical influences on it. There are actually a lot of work to sort of dissect and correct for and straighten out. But the better you get at correcting for all of that, the more you see that what's left is a real signal about the actual differences in the actual copy number of different loci. And so read depth, local variation read depth also carries information about structural variation at this locus. And Bob Hansiker here at the Broad has developed a set of analytical tools for really capturing all of these forms of information that are present in whole genome sequence data, and then bringing them in an integrated analytical framework to sort of develop a kind of maximally powered way to find structural variation of all sizes. And the sort of other kind of innovation in this is paying attention to all of the samples in a sequenced cohort at the same time. And just to give you a taste of what it's possible to do, this histogram and the red bars show the distribution of sizes of copy number variants that were ascertainable by array-based platforms, and you can see kind of a peak down around 10 kilobases. But you can see that with Bob's approach, there's in the same samples now, it has made visible a far larger number of much smaller structural variants, and that this approach relative to Bob's work to sort of bring all of these different kinds of information to bear on the analysis has actually resulted in a set of algorithms that have a far lower false discovery rate than any of the other algorithms that are available in the field, which I think is going to begin to make possible the stream of like a really rigorous, well-controlled association analysis at genome scale. That also compares favorably in sensitivity to all the other algorithms in the field, and that finally they can assemble the precise molecular sequence of each allele, exactly where the breakpoint is, and the sequence on each side. And so there's still not a lot of whole genome sequence data in the field, but this is sort of preparing for a future in which hopefully we will be drenched in these kinds of data sets, and it will make it possible to really fully utilize structural variation of all shapes and sizes to learn about the genetic basis of disease risk. So finally, I want to thank a lot of people who contributed figures and slides to this talk, in particular Bob Hansiker, Josh Korn, Mark Daly, Sean Purcell, and Charles Lee. Any other questions or anything people want to ask before we break for MPG? Yeah.
How do you validate those large deletions? Do you validate them?
So the most common way to validate them is just with real-time PCR, ideally with dual-labeled TACMAN probes. And those really large events tend to validate fairly well.